We are the Integrated Bionic Microsystems Laboratory, our iBionicS lab at the Department of Electrical and Computer Engineering. We work at the intersection of electronics and biology, with bioelectronic applications starting from wearable devices for humans to dogs, all the way to plant health monitoring.
To begin with, I'm going to be talking about how we collect data for precision agriculture and phenotyping and how the development of low-cost sensors could revolutionize these two fields. Finally, I'll conclude with looking at a new bioimpedance metric for quantifying physiological changes that I think could have a great impact on precision agriculture and phenotyping. I think there's three specific areas that field-deployed sensors have an advantage. First, there's certain fine peculiarities that you can only get in the field. Second aspect is that a lot of our remote sensing methods currently are limited by the weather, and so with field-deployed sensors we can understand what's happening in real time. With precision agriculture, these are relevant because it allows for the specific application of resources in a wise and conservative manner. And with phenotyping, the additional data helps us better understand the various effects on the expressions. We started the journey of creating our own system. Our system is housed within a commercial enclosure with venting to allow for airflow to pass through it. There can be 1 or 2 2.5 watt solar panels on top of the system to allow for charging and powering the system throughout the day. But at night, the battery provides power. The charging and regulation is taken care of on the main board that also includes a LoRa transceiver as well as an ARM microprocessor. This collects the data from the various front ends, which includes sensors for light, soil moisture, temperature, humidity, and most importantly, bioimpedance. Realizing that there was a connection between the water status of the plant and bioimpedance, we conducted an experiment where we wrapped soybean plant pots in plastic to inhibit water loss to transpiration only. The bioimpedance shown in the graph above increased for the drought group. A key takeaway is that the bioimpedance rapidly increases and is sensitive to water values. It tells us exactly at what point a plant will respond to differing water values. Another technique that we've been developing that could have widespread consequences are the study of electrical potentials in plants. These potentials are caused by the movement of ions in the plant tissue, creating an electrical signal that is similar to that of the human nervous system. We measure these using some state-of-the-art electrodes that we can use to measure the potential changes on the plant leaves. They allow the plant to essentially breathe through the electrodes and don't damage the leaves themselves. The hope is that one day we can use this to our advantage, where we can detect in real time when a plant is under attack by insects or when it is experiencing harsh conditions. This can allow for farmers and scientists to develop better methodologies to help protect the plants, as well as discover plants that are more resilient and resistant in the face of climate change.